Hello, this is the third part for Chapter 3, and in this part of Chapter 3, we are going to describe translation. There we go. Translation and transcription. Transcription comes first, translation next, they're alphabetical. Transcription is the DNA coding and making the messenger RNA, and translation is going from the messenger RNA, leaving the nucleus, to going out and creating a protein. You really only need to know the players and what they basically do, but for me, to understand the bits, it helps to understand the whole process. So I'm going to talk briefly about the whole process so you get an idea of what's actually going on. And to do that, I've got some of my favorite videos. So let's look at this video. Okay, so in this video, we are going to, let me describe what you're seeing here. So first off, this coiled pink thing is the DNA, and these are the enzymes that will break it apart. I will keep the captions on. Notice that sometimes the spelling isn't good, but just in case you have difficulty hearing the sound, even though I do have the sound turned up, it's a little tricky to hear. Okay.
Let's begin. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand.
Okay, let's go through and look at what's happening here. So if we go back to here, what you have is this pink is the DNA, and this big blobby colored thing, these are the enzymes. It's very important you realize these things don't just happen. You have to have enzymes and controllers to make them happen. So here's the enzyme, and here's the controller here. Then once it all starts, you end up with this blob, which is the blue blob, which is, again, enzymes. And at this point in time, you don't need to know the specific enzymes. You just need to know their enzymes. So the blue blob will then go and read the DNA. So the blue blob will break apart the DNA, and it reads one side. And here you can see these are the nucleotides, or the parts of the DNA and RNA, or the RNA molecule. So as it comes in, they will be matched up with the opposite code. If you remember A to U, because there's no thymine, A to U, C to G, T to A, and this is how it will go through. So the enzyme, the polymerase, anytime you see the ending ASE, that is an enzyme. So the enzyme will unzip the DNA, match up the nucleotides, and that will produce the messenger RNA, which you see here. So enzymes control it all, enzymes wield it together, and it is actually quite fast once you get started. So here you can see, almost, here you can see the enzyme will then weld it together, and this yellow thing at the end is the messenger
of an
RNA.
RNA molecule, which is assembled one subunit at a time.
Okay, so here is the messenger RNA. The DNA is a very large molecule, so it will not leave the nucleus. The messenger RNA is the copy of only a certain part of the DNA, not the whole thing, and it usually just copies one specific gene, which is the material for one specific protein.
By matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Okay, very important you realize this is a complex, very complex system. It's not just going to happen. You've got enzymes, you've got little molecules that feed things in, and then each one of these nucleotides will build this messenger RNA, and that is the function of transcription. Now we're going to continue on with the second process, translation. So here is the messenger RNA that has been created inside the nucleus.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for.
So when you look at this diagram, let me associate it. Here is the nucleus, and these are the nuclear pores. So the messenger RNA has to come out of a certain area. Then the messenger RNA comes into the cytoplasm.
There can be several million ribosomes in a typical eukaryotic cell.
So here we need the extra organelle. This is the ribosome, and the ribosome acts as a workplace or a workbench. Along comes the messenger RNA, and it is put into a ribosome.
These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape.
Okay, if you remember, the messenger RNA has these base pairs, the A's, C's, G's, and U's, and they're going to be read 3 at a time, and each 3 is called a codon.
The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs.
Okay, so if we look at the transfer RNAs, you've got the little red dot, which is where the amino acid is, and then the transfer RNA is what transfers the amino acid and matches it up with the 3-letter code. But on this one, the 3-letter code is the matching code, so this is called the anticodon. And the whole thing is called a transfer RNA.
That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the...
Okay, again, very complicated, but I just want you to get the general gist. So in the messenger RNA is fed in through the ribosome, and the ribosome has space for 3 different transfer RNA. The first one comes in, and it will then donate its amino acid, and then the amino acids are using enzymes welded together to make peptide bonds, and then this amino acid chain becomes the peptide or the protein chain, and it leaves, and then the transfer RNAs work through the ribosome and end up going back into the cytoplasm, getting new amino acids and recycling.
The ribosome removes each amino acid and joins it onto the glowing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence.
Okay, so the code starts in the DNA. It's read and copied by the messenger RNA. The messenger RNA comes out, lands in the ribosome, and the ribosome is the workbench that puts things together. Along will come the transfer RNA, and the transfer RNA carries the amino acids in a specific order based on the code, or a specific amino acid based on the code, and then this will build together in a chain, and then this is the protein. And then the protein, once it's done, will leave the ribosome and go to wherever it's needed. Often it goes into the Golgi, into the endoplasmic reticulum to be modified and changed, and then into the Golgi, and then transported out with a vesicle. So, although this is a very complicated process, including all sorts of enzymes and details, I just want you to know the general idea of what is going on and a general idea of the players, as I call them. So DNA, messenger RNA, ribosome, transfer RNA, amino acids, proteins. Okay, so that describes the process of transcription and translation. Another process that's very important in the cells is mitosis and meiosis. The next process I want to look at is the process of mitosis. Now, mitosis is the process of duplicating cells and making the exact same cells as your starting cells. So your parent is the exact same genetic material as your child. Now, it is very complicated, has lots of stages. I want you to just understand the general gist of what's going on. So here is the chromosomes of the cell. Mitosis is the orderly division of these chromosomes. So if we look at it, here are the chromosomes. Before they go into the process of mitosis, the chromosomes will duplicate. Then you have a double copy. Then as it goes through the process of mitosis, what they do is each chromosome will coil up so you can see it under a microscope. Then they line up, and then one of each copy is pulled to the pole of the cell, and then the cells themselves will divide the cytoplasm. And this was a plant cell, and you can tell it's a plant cell because this is what's called a mitotic plate. So now you have two different cells. So that's looking at in terms of chromosomes under a light microscope. If we look at it here, they've stained it. So here you can see your cells, and then your cells have your chromosomes dividing. And that's pretty much what I need you to know. The process of mitosis is cell division, starting with one parent and resulting in two daughter cells that are genetically exact to the parent cell. Okay, now let's briefly look at the process of meiosis. In meiosis, this happens in reproductive cells, and so that means sperm and egg. So here is the egg. Look how large it is compared to the sperm.
Meiosis is a special type of cell division necessary for sexual reproduction. These are sperm-producing cells of a male crane fly, a species with only 8 chromosomes per cell. During meiosis, the cell divides twice, producing 4 haploid cells.
So, hopefully you can see it with the resolution. Here is the chromosomes, and this is the cytoplasm around it.
Containing 1 copy of each chromosome. The first meiosis division separates pairs of homologous chromosomes, creating 2 haploid cells with only 1 set of chromosomes. The second division separates the chromatids in each chromosome. The end result is production of 4 haploid cells, each with a single chromatid from each chromosome.
Okay, so let's compare the overall process. With meiosis, you start almost the same as mitosis. You have a duplication of the chromosome material. There is some what's called crossing over, which allows for a little bit of mixing. Then you have 2 divisions. So it divides, separating the chromosomes and then separating the exact copies. And what you end up with in this situation is 4 cells, and each of these cells is genetically different, and each of these cells contains half the chromosome numbers of the parent cell. So now you have, in this case, sperm formed, and then these sperm can be packaged, these chromosomes can be packaged, given a tail, and you have sperm. When we're comparing the process of mitosis and meiosis, here's a really good summary. So they start the same. A human cell has 46 chromosomes, or 23 pairs. So in mitosis, you divide once and you end up with the exact same number of chromosomes as you do in the parent. In meiosis, you divide and then you divide again, and you end up with half the chromosomes as in the parent. Now, these processes have many, many steps, but this is enough summary for what you need to know at this time. Okay? And that is the end of Chapter 3.